Hello, everyone. Good morning and welcome to this webinar on Good Friday. Usually most of us should be on vacation on this Easter Friday. Instead, we're all sitting at home and listening to a coronavirus lecture. And this is, of course, due to the current global crisis that we are all in. And I was asked to give this presentation on coronaviruses. And you might wonder why me. I'm an assistant professor at the Department of Life Science and Medicine at the University of Luxembourg. And although I normally do cancer research with a focus on melanoma, I have a formal background in virology. I actually did my PhD in virology on measles viruses. So this kind of qualifies me to give you this lecture, and I also do give a virology lecture to the biology students, the bachelor biology students at the University of Luxembourg every year. So you were sent an outline for this presentation today, which I changed a little bit. You will see here. So I kind of shorten it. Let me try and get the pointer here. So here it is. I've shortened the outline a little bit for the sake of time. The idea of this lecture is to give you a broad overview and also give you some detailed information about the biology of the virus, because I suppose until, let's say, 2 months ago, most of you were probably not very interested in virology. And over the course of this crisis, many of us have become very interested in the biology of viruses. So we're going to start with a super brief introduction into virology. This is normally taking 20 hours in the normal lecture. I'll try and condense it to 10 minutes. So what are viruses? Here's the formal definition. Viruses are submicroscopic, obligate intracellular parasites. And most importantly, they don't have an own metabolism. So they're fully dependent on the host cell, which can be humans, animals, plants, bacteria, anything you can think of. They come in different flavors. The nucleic acid, the genetic information might be DNA or RNA, which is always surrounded by a protein coat, usually called the nucleocapsid. Viruses may have an envelope or not. In the case of SARS coronavirus 2, we do have an envelope. The International Committee of Taxonomy of Viruses, abbreviated ICTV, is taking care of the classification of viruses. So we currently have far more than 8,000 different viruses, and the classification is quite a complex thing to do. Now, let's briefly look at the key steps of a viral life cycle. So a virus, generally all viruses, start by attaching to a host cell, and this process is called attachment or adsorption. And it's always, or generally, or 90% of the cases, done via a receptor surface protein interaction. This is followed by penetration of the virus. So the virus is taken up by various mechanisms into the cytoplasm of the host cell, where it then uncoats. So the protein shell of surrounding the nucleic acids are broken up. The nucleic acid is released into the cytoplasm. This is then followed by a biosynthesis or replication part, where the nucleic acid is replicated, and also the proteins are synthesized, which are required to generate progeny virus. This is then, in a pre-final step, assembled again in the cytoplasm of the cell. So mature virus particles are formed inside the cell, and then often upon release of the cell, there's a final maturation process of conformational changes of surface proteins, and this then releases thousands, 10,000s, 100,000s of mature viral particles into the surrounding. All right, and now viruses, as I said before, come in different flavors. We know DNA viruses, RNA viruses, and retrotranscribing viruses. So DNA viruses, and there are grouped into 7 classes according to the architecture of the genome. And the coronaviruses are here located in group 4, and they have a single-stranded RNA genome of positive polarity. And I'll come back to the positive polarity in a few minutes. Now, another famous group that you all know are the single-stranded RNA viruses in group 6, and they contain, for instance, HIV. Measles virus, on the other hand, most of you know as well, is part of group 5. So this classification is very important because the biology of these viruses is highly distinct according to the genome. Viruses are then also grouped not only according to the genome, but also whether they have an envelope or not, according to their size, the capsid symmetry, it has an architectural symmetry, and the mode of transmission and the host cell. And all this is put together into a classification system. Now, given the current pandemic and the global crisis that we're facing, it's always good to have a look at historic events and look at previous epidemics and pandemics to put things into perspective. And I just added a few here for you to see. Let me just see whether you can see my pointer. I hope you can. So we have only one great epidemic in the 1500s, which was called by DNA viruses, smallpox. And it's in fact the only virus that we've ever managed to eradicate from the globe. All other epidemics in the past or recent are caused by RNA viruses. And most notably by influenza, various influenza types, by Ebola, and now in the last 20 years by coronaviruses, SARS, MERS, and SARS CoV2. Now, why is that? I mean, this is a striking observation that all these new epidemics are caused by RNA viruses. And there's a reason to it. RNA viruses are much more variable. They have an incredibly high mutation rate, many of them. And this is because the viral RNA polymerase that is used to replicate the RNA genome has no proofreading capability. And this is in contrast to the DNA polymerases that can go back and repair a nucleotide that has been erroneously incorporated during replication. Now, RNA viruses cannot do that. So over their generations, they accumulate many mutations, a lot of which are fatal, some of which are beneficial to the virus. It's also notoriously difficult to make vaccine against these viruses because of the high variability. Now, let's have a look at coronaviruses. Now, this is a very simplified overview, which I selected because it was published in The Economist, one of the largest economic newspapers in the world. It was on the title page. And if a virus is shown on the title page of The Economist, you know that the world is in for trouble. So it's actually the most important things that you need to know are the proteins shown here. You have a membrane protein, you have the spike protein, which we'll talk about in a bit more detail, the nuclear capsid surrounding the RNA, and an envelope, of course. And what else do we have? The nuclear protein I mentioned, membrane protein, right. So this is the most important things we have to keep in mind. The taxonomy of coronaviruses looks like this. The order is the Nidovirales. We don't have to go into the detail here, but we have 4, let's say, subgroups of coronaviruses: the alpha, the beta, the gamma, and the delta. Only alpha and beta viruses are important for us in mammals, and the others have so far not been found in mammals. Now, the first coronavirus to attract our attention caused an outbreak in Asia in 2003, and this one was called SARS CoV, because it was the first one. And more recently, 8 years ago, there was another smaller outbreak, but with a very high case fatality rate, which was named Middle East Respiratory Syndrome virus, MERS. And that has interestingly jumped from camels onto humans. And now we all know the current epidemic has been caused by COVID-19, also called SARS CoV2. And from December onwards last year, it has been successfully spreading the world. Now, phylogenetically, or the molecular epidemiological studies that are currently undertaken are basically extracting the nucleic acid of the virus and then sequencing the RNA and aligning it using phylogenetic programs. And this is all, I mean, all the publications and figures that I'm showing you are from papers that have been published over the last 4 to 6 weeks, which is quite remarkable. So you see here that our current SARS coronavirus is very similar to the MERS and to the previous and other human coronaviruses. There are a whole number of different species, but they all look alike. The life cycle of the current virus that we are dealing with looks like this, and this is interesting to understand, because if we want to develop therapies, we need to understand the biology of the virus in order to develop inhibitors or interventions for the specific steps. So we start with the virus attaching using its spike protein, here shown. I hope you can see my pointer, I'm not sure, to the ACE2 receptor. And I'll mention, or I'll give you some information on this receptor in a second. Then there's the engulfment. So the virus is taken up by the host cell, and this process is completely unclear. We don't know exactly how this happens yet. There's a lot of research going into this part. The positive sense polarity RNA is released into the cytoplasm, and I mentioned before, I'll come back to it. So here's the positive polarity again. And what that means is that the positive polarity actually equals mRNA architecture. So this RNA is almost like a human mRNA. It can be immediately translated into protein without further ado. So this RNA is then hijacking the host cell's translational machinery, the ribosomes and start factors, transcription factors, and everything else that is required in order to transcribe the RNA into protein. The first protein that is transcribed is the viral polymerase, which is called RNA-dependent RNA polymerase, RDRP. And this one is required because, or the virus has to bring the information for this enzyme, him or itself, because we don't have this enzyme in our cell. Then this viral polymerase copies the positive polarity RNA into negative sense RNA. This negative sense RNA in turn serves as a template to make subgenomic RNAs, which encode for the different structural proteins of the virus, and also to serve as a template to replicate the genome. Because we need all these individual parts. We need to make many copies of this RNA to be packed in the mature variants, and we need to make all these proteins that we require to make the shell and the spikes, etc. So this is done in the cytoplasm, and then the virus is maturing and eventually released. A lot of the details of this process are, of course, still unclear because we're looking at a very new virus. And this is a short form of the life cycle that I will skip now. It's just a short version of what I've just shown you before. Now, let's look a little bit at the biology and some key facts of SARS coronavirus, or the current knowledge of what we have. So here again is the genome. Coronaviruses notably are the largest RNA viruses that we currently know. And this is important because that makes certain vaccine developments very, very difficult, because it's for human genome comparisons, it's tiny, but for viral genome, it's rather large. So this one is roughly 30,000 nucleotides. It comes in 2 open reading frames that encode for non-structural proteins. Here our RNA polymerase that I mentioned before, some proteases that are required to slice the polyproteins into smaller proteins, and then everything that is required for the envelope. What's also something that is very favorable for this virus, or very clever of the virus, it has a 5' cap structure and a 3' poly A tail. And you might remember from your biology lessons, this is protecting the RNA from internal RNases. So the genome of the virus upon entry in the cell is really nicely protected. This is something I just added for those who are will be interested in more detail to come back to. It's a nice picture from a Nature publication, from a Nature review, where there is a comparison between MERS and SARS. A very similar thing I showed you before. So the genome organization is always the same and very, very similar between the different coronaviruses. Now, to focus a little bit on the spike protein, because it makes the contact with the receptor, it's of course very important to know the sequence, and new viruses are currently being sequenced all over the world. And I apologize, this is a bit blurry, but you will see here at the bottom, on top we have a human SARS CoV2 virus, then below an isolate from the bat, and beneath a pangolin. And I'll come back to the pangolin in a second. It's a funny Asian animal. And just look at the amino acids that are similar between the human SARS CoV2 and the pangolin. And this is quite a remarkable similarity, and this is why people think the pangolin plays an important role here. Now, the spike protein of the virus comes in 2 different conformations, a closed conformation, and it needs to be open conformation in order to make contact to the ACE, to the angiotensin II converting enzyme, which is an important enzyme for regulation of our blood pressure. Now, this is a detail that was just published, I think, 3 days ago, quite interesting. The mechanism, so a spike makes contact with the IS2 receptor, and then we have the serum protease with a fantastic name of TMPRSS2, which needs to be in close proximity, and this protease then slices the spike protein or cuts it, and only this slicing then activates a fully functional entry process. And very recent results on blocking this serine protease shows that entry is actually slowed down, and the entry of the virus will be less efficient if this step is not occurring. And the virus can still get in, but it's less efficient. So this, of course, would be a target for therapy. All right. Now, where does this virus come from? This is something, a very important term in virology is tropism. And we have 3 levels of tropism: the cellular tropism, the tissue tropism, and a host tropism. So what we know for sure is that coronaviruses come from bats, and not only coronaviruses, but also, for instance, Ebola. So bat is not a very good animal to have around you. From the bat, the viruses can jump species, and this has been shown before for Ebola. And here, for the current pandemic, the idea is, or the current knowledge, that from the bat, it jumped to this pangolin. Here it is. It's a funny-looking animal that is common in China, and this connection was made because of the sequence similarity of the spike protein. However, I read yesterday that there is a little bit of doubt now whether it's really the pangolin or something else. Anyway, from the pangolin, I don't know how closely the Chinese live together with their pangolins. It has managed to jump into humans. And from within a human, we need to look at which kind of tissue and which cell types can be infected. And unfortunately, it has a broad tropism, so it can infect many different tissues. And by using this AC2 receptor, which is ubiquitously expressed on endothelial cells and preferably also on pneumocytes, of course, it can get into many different cells, which doesn't make life easier if it comes to treatment. Now, this tropism that I just showed you before makes SARS CoV2 a textbook emergent pathogen after species jump. This is the correct term in virology. Now, for these kind of viruses, like I said, coronavirus is not the first one to do this. Two scenarios are possible. We have viruses with little capacity to spread from human to human, so low transmission, but the case fatality rate, CFR here, could still potentially be very high. Then we have the second group, which acquires further mutations and generally seen in RNA virus. And these mutations, if we are unlucky, if humans are unlucky, equip the virus for better human-to-human transmission. And this is exactly what we see here. So we have the virus has made a successful trade-off. It has become less virulent, but more transmissible via droplet and aerosol. It is also causing a fairly mild disease, so the majority of infected people is not killed by the virus. So the viruses that fall into this group, they in principle carry the recipe for disaster. And this is what happened. Potentially a pandemic can evolve. We have the stochastic species jump. We have transmission to and then in between humans. We have a local epidemic thereafter with a high number of asymptomatic cases, a relatively mild disease, and a very high transmission rate. And from there on, and given our global extensive traveling mode, we soon develop, or we potentially develop into a pandemic. And this is exactly what happened here. Right. So where are we now? These are numbers that are from Worldometer from this morning in comparison to yesterday. So we are currently at 1.6 million confirmed corona cases with close to 100,000 related deaths, which brings us to a case fatality rate of almost 6%. This case fatality rate, once this pandemic is over and we have better numbers, will be much lower. That is for sure. I'll give you some evidence for this in a minute. An important point here is also that the vast majority of cases is currently in mild condition and only, luckily, 4% are critically ill. But given the overall tremendous number, of course, we have a large number of people who are seriously ill, and now with coronavirus hitting Africa and hitting Asia and some other parts in Asia and Southern America, we have, of course, many more critically ill people who are probably not registered yet. So I put together a couple of numbers that I think are worth while looking at, focusing on our direct neighbors here, Germany, France, Belgium, and this is the current toll for Luxembourg. We have 3,115 cases today. Yesterday we had 3,034. And we have the highest number of cases per million of population in the world. If you look at the Worldometer table, this is really the highest number, 4,850, even higher than Iceland. Now, why is that? We are a very small country with only roughly 600,000 inhabitants, and we do a lot of tests, also shown here. Only Iceland and some other countries are doing more testing than we are, so this is a very good effort here. And that brings us to, and Luxembourg being a very international community, this brings us to a relatively high percentage of positive people in the population. Now, looking at the cases, the daily increase in cases in a comparison of Germany and Luxembourg, you might think that this is a Gaussian curve that is almost approaching to normal levels again, but I think it might be a bit too early to claim this. So I guess we still have to be patient on that one. Now, an interesting comparison and a very important comparison we need to do in this pandemic is looking at case fatality rates. And this is something that is really striking and that I saw this morning also. As of today, France and Germany have exactly the same number of cases. This is France, this is Germany. But France has an almost 5 times higher case fatality rate than Germany. And this is really something for politicians, healthcare professionals to figure out after the pandemic what causes this huge difference here. Whereas we can assume that the virus is pretty much the same in France and Germany. So there are several reasons that we know of, but a lot of things need to be figured out after this epidemic is over. Now, why do we have such huge differences in case fatality rates if you compare, for instance, Italy with China? And there are more and more studies coming in looking at the epidemiological pictures. For instance, Italians are in general much older than the Chinese. And then, of course, when the epidemic started in Italy, they only tested in the first few weeks, they only tested the hospitalized people. So of course, you have a much higher case fatality rate because you're only testing the very ill people that already made it into the intensive care unit. These numbers will drop for sure. And this is also something interesting in really determining the case fatality rate, the number of critical care beds per 100,000 of population. And you see here Germany and Luxembourg being right on top. And this is why probably we see fairly low case fatality rates in those countries, luckily. The overall characteristics of the case fatality rates are summarized in this nice paper that came out 2 days ago in Pathogens. So we see, of course, the trouble appearing in elderly patients, really hardly any problems in the very young population. In children, almost absolutely no fatalities whatsoever. And interestingly, we see that 99% of cases have a predisposition. They have a chronic disease, and some of them are listed here. So it's really, I think, by 3-4 months into the crisis, one can say that people who die from this virus are preferentially the elderly or people with a previous health condition. So some key facts, those are assembled from the Robert Koch Institute in Germany. They update this regularly and have nice summaries. What do we know about this virus until now? We have a droplet infection, mostly. We do have smear infections and aerosol infection, however, to a much lower degree. The median age of infected people is around 45, and we have the R0 number of 2 to 3. And I guess you've all heard about this famous R0 number in the news over the last few weeks. It's the reproduction number. And just here on the right, I'll use my pointer again, you see that the R0 number below 1 means you cannot have an epidemic. And that means that a positive individual can infect less than 1 susceptible individuals around them. R0 greater 1, epidemic becomes likely or possible. And just as a comparison, the Spanish flu in the last millennium had an R0 rate, which is very comparative of our SARS coronavirus here, 2.3. Tuberculosis, on the other hand, has a reproduction rate of 10, and the highest reproduction rate that we know in virology comes by measles viruses with up to 18. So one infected individual can potentially infect 18 individuals if they are susceptible, not vaccinated, or haven't had previous disease. What else is important here? We have infectious period 4 to 5 days after symptoms appear. And there's more and more evidence coming in that people are symptomatic or can be infectious long before symptoms appear. And this is making the control of this virus much more problematic. I mentioned this before. Yes. And let's see what we have here, right? Tenacity. This is basically measures the viability of the virus in aerosols or various surfaces. And this is the only study that is so far available on tenacity from the New England Journal of Medicine. And we need to get more information on how long the virus actually survives, and not only whether we can have infectious particles on, let's say, door handles and whatever kind of surfaces that you are in contact with. But the key point will be how many infectious particles survive. So in laboratory conditions, of course, we can measure infectious particles, but would they be sufficient to really account for a large number of new infections? Probably not. These numbers, however, will have to be updated, and the virologists or epidemiologists are urgently waiting for new information on tenacity numbers. A key role in this whole pandemic, and this is what we are beginning to understand now, is the asymptomatics. So people that have absolutely no symptom of disease. They have no idea that they are infected, however, they are infected, and they carry the virus, and they can be infectious to others. So there is more and more global effort going into figuring out how many asymptomatic cases we have in our population. And there is a recent studies by Jeffrey Schaman's group who showed that almost 55% of documented infections could be due to asymptomatic carriers. Now, what that means is it's good news, because if we have a large proportion of asymptomatic carriers, that means it drops our case fatality rate tremendously. And this is exactly what will be the case. It also means good news because we have a higher level of immunity in the population. The bad news, of course, is that our flatten the curve measures might, and the models that we are calculating to suppress the viral spread might be optimistic if we have a large proportion of asymptomatic carriers. In connection to this, there is recently a prevalence study has been launched in Luxembourg, or it's starting soon. It's called CONVINCE. And this is exactly looking at this proportion of asymptomatic carriers, assembling 1,500 randomly selected participants over a longer period of time to look at the presence of the virus, and I think later on also at the seroconversion, so whether people have already developed antibodies and a protective immunity against the virus. Very new data from Germany, actually yesterday, is a similar study that has been conducted in Heinsberg. It's a place in Germany in North Rhine-Westphalia where the German epidemic has actually started. And this was, I think, in February during Carnival. And there was an infected couple that attended a huge carnival get together with, I don't know, thousands of people or hundreds of people. And from there on, this village or the city saw an explosion of cases. And now they went back into this village and assembled also 1,500 people. And yesterday they released the preliminary data on 500 of those people. And interestingly, they didn't only look for the virus itself by qPCR, but also serology. They found in the asymptomatic, presumably healthy population, 50% positive and immune individuals. So those people were IgG positive. The claimed specificity of the test was larger than 99%, which is quite good until now. But overall, this herd immunity is far too low. I'll come back to the herd immunity in a second. Now, with this, and the striking number is that the mortality, the case fatality rate in this region, although there was a huge outbreak with large case numbers, 0.37 compared to the 1.9% reported by the Johns Hopkins University for the entire Germany, not considering the healthy population. So this is probably closer to the realistic, this is a far more realistic case fatality rate that we will see in other parts of the world as well. And it might be very similar in Luxembourg, probably even lower. What has also come out from this study is that the number of infecting particles seems to be very important for the severity. That means the more particles you are taken up by a droplet, by speaking to someone who is infected, or by direct contact, close contact with an infected individual, the number of virus particles that you are ingesting into your system, into your through the mouth or the eyes or the nose, is key to the severity. The fewer particles you take up, the milder the disease. This also means hygiene measures are really very important to continue to do, because they reduce the number of particles. So if you wash your hands regularly, you might not get rid of each and every single particle, which is probably not necessary, because you can get rid of most of them, and that is very important. The immunity might last 6 to 18 months, they conclude from the study. But this is probably early days, and we'll have to wait and see. And the study also recommends that in Germany, slowly but surely, the phase 2 of the German pandemic plan can start, and that means relaxing quarantine measures, slowly but surely. But listening to Chancellor Merkel yesterday, she was not very keen on doing this, so we continue with our containment measures. As I said, herd immunity is a term that you've probably heard a lot about in recent weeks. So that means the percentage of individuals in a population that are immune to a given disease. And because SARS-CoV2 is new, nobody was immune from the beginning. And we need to get to a herd immunity of 60–70% of seroconverted individuals in order to protect the entire population from further outbreaks. And this can be done either at the moment by social distancing, because we don't have a vaccine or treatment, so we need to do it that way. Here's my cursor. Or eventually by immunization, so vaccination, and try and make the population or protect the population. And yes, this is what we are hopefully going to see only next year, probably not this year. I was told that there were already lectures on epidemiology, so I'm not going to spend much time on the basic principles of epidemiology. I have some extra slides if you're interested. But the general consensus in Europe was that we follow the flatten the curve policy. So we're trying to slow down the increase of the number of cases in order to gain time until we have a vaccine and better treatments available. And quite a nice quote by Professor Sansonetti from the Institut Pasteur in Paris. He said that statistically speaking, the 1% mortality that we are seeing in SARS-CoV-2 probably lower at the end of the day, and the 10% severe cases with less than 5% of people needing ICU is probably not very worrying. But given the overall numbers, transmissibility, and infectivity of the virus, this is obviously putting our healthcare systems into dire straits. And this is what legitimizes this very severe flatten the curve policy, which we all know consists of containing the spread of virus by social distancing. We will have to build up sufficient herd immunity. We need to increase the testing, the virus itself, and the antibodies against it, so to see how big the proportion of protected individuals is in the population. We will see a much larger use of personal protection, masks, personal hygiene, washing hands will continue to be very important for a long time to come. We will hopefully soon be able to move from social distancing to smart distancing, so allow certain activities and really make sure we are still keeping distance between each other. And there might also be the introduction of contact apps to be downloaded on our smartphones that can then follow up or identify and notify contacts of potentially positive individuals. Right, here's this contact app. I'm not going to go into it, but here's the publication by Ferretti et al. And they have really looked at what such a contact app could do, looked at the pros and cons. I guess it will come, it very much looks like it. Whether it's going to be as or has as promising effects as some people claim, we will have to see. But it might help to reduce the overall spread of the virus, and therefore I think it's legitimate. Now, looking at or being in the situation that we are currently in, all of us in home offices, in containment, with everyone knows positive cases of coronavirus now, some mild, some severe. It's always worth to look back and think about some of the other epidemics, and epidemiologists all over the world are currently doing this. And the classic pandemic that is always worth looking at is the Spanish flu 1918. And I put this up here for you if you're interested to read this. But this is the key picture or graph that I wanted to show you. The Spanish flu came in 3 waves. Now, not that I want to scare anyone, but it's very, very likely that we are going to have a second wave of SARS corona and possibly a third, hopefully much smaller wave. Now, for the Spanish flu, you see that the second wave was by far the largest one. But with the current technical advances and development that we are having, this is hopefully not going to be the case for SARS corona, because it is hoped that by next year we will have a vaccine, and then the second wave might hopefully be much smaller. Now, also worth looking at or comparing COVID-19 or SARS coronavirus, which is here, to some other viruses, is by comparing the reproduction number that I explained to you before, and put this into context of the case fatality rate. And you see that smallpox, the only virus that we've so far eradicated, really takes the lead here. The MERS was highly fatal, but luckily only caused a few cases. And Ebola, as we all know, has incredibly high fatality rates, going up to even 30% in some outbreaks. And one of the top scoring viruses here is measles, with its incredibly high case fatality, reproduction number, sorry. And so COVID is kind of in the middle, in between the influenzas, the measles, and the Ebolas here. Right, and in direct comparison of SARS coronavirus with the other SARS viruses and with flu, some interesting number crunching here. And the reproduction number we talked about, the case fatality rate, if you look at this, of the SARS outbreak in 2002 in Asia, was around about 10% compared to, this is not the real number, but likely much lower. For MERS, it was almost 35%, so really large number, high case fatality rate, but luckily only very few cases. For influenza, for instance, and this is the big comparison that we always use these days, we have much larger numbers of infected people worldwide per year. We have, however, we have probably a slightly lower mortality rate for influenza. Could also be at the end of the day that we come up with a very similar mortality rate. Some people are already wondering whether this could be the case. Now, with this, let's have a look at how we are diagnosing SARS coronavirus. What are the current updates on treatment possibilities, and how far are we on the vaccine side? So SARS luckily can be isolated quite easily in tissue culture. We have many susceptible cell lines available for laboratory isolation, and the virus is causing classic cytopathic effects, which can be seen here. So this is an uninfected tissue culture, and this is what it looks like after 24 hours of SARS infection. So you have the classical pictures of a viral infection in tissue culture. This is a summary slide from venture capital, a company called Artis, and they've nicely put together the current numbers on where we are in diagnostics and also treatments. So we have 42 commercially available PCR tests and 2 next-generation sequencing platforms. And the tests that we are currently having, the majority of them takes 2 to 8 hours to run, and only 8 of them are completed within less than 1 hour. So, of course, what we would like to have is highly specific and sensitive tests that are very quickly. But there's fierce development going on in this field as well. Now, what are we doing right now to diagnose viral infection? As you all know, a PCR is performed. So there's a throat swab or a nasal swab, and then RNA is isolated from this swab, and there's a reverse transcriptase PCR performed on the viral RNA, using a set of primers specific against generally 2 to 3 different genes, the nucleocapsid gene, the polymerase, etc. And so different genes in order to make sure we have a high enough specificity. And the nice thing about PCR is it's semi-quantitative, so the results are also a proxy for the overall sample viral load. So from the results of the RT-PCR, we can get an indication of how much virus is present in this in a patient. On the other hand, we urgently need serological tests. They will obviously be performed on blood samples, and they are much more suitable to get an idea about the timing of the infection. So whether we are looking at the recent infection past, and also the overall prevalence of immunity in the population. There are tests out there, and they basically come out on a daily basis. However, they are, to my knowledge, not available to the extent that one could really order 100,000 tests until to be delivered next week. So the companies are still in production problems to provide as many tests as are being requested by global demand. But of course, serological tests, generally done in an ELISA format, if it comes to a diagnostic lab, are capable of detecting or measuring IgM antibodies and IgG antibodies. An IgM, being the first antibody class appearing after infection, indicate a recent infection. And once we have a higher level of IgG antibodies, that would imply that the infection is at least 4 weeks ago, and that full immunity has developed in that particular patient. All serological tests are measured against RT-PCR test. So this is the reference test, and whatever the specificity and sensitivity of the serological tests will have to be measured and evaluated against the PCR. And what is important is there are a couple of quotes from a very recent paper from The Lancet. And what we really need to be careful about for serological tests is the specificity. Why? We had previously coronaviruses, and very harmless coronaviruses, circulating in our population, and we currently have them all around us. But they are very mild. They either give you no disease or very mild cold, and most of the antibodies or the antibody tests could be, or some of them are cross-reactive. So it could be that we are picking up antibodies against these milder brothers and sisters of SARS CoV2, and this is, of course, not what we want. We want specific antibodies against this particular pandemic virus and not the other ones. So specificity is an issue, sensitivity as well. Very important, when are we sampling, and how high are the titers of the antibodies at a certain time point of sampling. So we need longitudinal sampling and get a few blood samples from patients over a couple of weeks or months in order to get a real idea of what is happening in the population. And the WHO, a few days ago, last week, actually has launched the biggest serology study mankind has ever seen. It's called Solidarity 2, and almost every country in the world is participating. And I assume that the Luxembourgish study on the asymptomatic prevalence of SARS coronavirus 2 in the population will also feed data into the Solidarity 2 study conducted by the WHO. Like I said, there are a number of tests that are out there already. Ideally, we will have a quick test shown here, which could almost be done at home. It's almost like a pregnancy test. You need to prick your finger, a little blood drop, and then wait for a few minutes, and you will see whether you have IgG, IgM, or no antibodies. There's a positive control and a negative control. So as far as I know, these samples are not available, or tests are not available yet into larger amounts, but I'm sure they will come soon. So I think I mentioned all these points. Yes, and then one last point here. We have different tests, different companies, different providers. They all have differences. And this is also going to be an important point. How comparable are the serology data that we get from these different tests? A test like this is generally much less sensitive than a lab-based, diagnostic lab-based ELISA test. So we need to make sure that what we are doing here is also nicely comparable to within a country and also compared to other countries. On the qPCR side, like I said, we start with a sample, do the sample preparation. So first the RNA has to be extracted, then we do a reverse transcriptase on the RNA and amplify parts of the nucleocapsid gene, the envelope gene, and this RDRP gene, the RNA-dependent RNA polymerase. Together with positive control amplifications in the same sample and negative control primers, this gives you a highly specific semi-quantitative result on whether a person is infected with the virus or not. Currently, there are 49 diagnostic kits in trials or available, 40 treatments being available, being tested currently, and this number is rising daily, and 5 vaccines are currently tested in clinical trials. So here's just a summary of how many tests or how many treatments are targeting what kind of symptom or what part of the viral infection. So this is treating symptoms is what we are mostly doing right now. Then anti-inflammation is something which is very important. And this is looking mostly at something called a cytokine storm, and a systemic inflammation that is induced by viral infection in some of the severe cases. Not all of the severe cases, but in some of them, we can see a cytokine storm, which is very difficult to treat and leading to severe complications, can lead to death as well. There is tocilizumab, which is an anti-IL-6 antibody that specifically targets exactly this systemic inflammation, and there are currently clinical trials going on using this antibody. Apart from this, there are different treatments in different stages of trials, and I put together a brief overview for you here. It's not comprehensive. There are others currently being tested, but those are the most important ones. Remdesivir, you've heard about it, is the adenosine analog, which was actually developed against Ebola. Some effects, it's highly disputed whether it really has a systematic effect or whether it was occasional. Chloroquine is an anti-malaria drug, which blocks the interaction between the surface, the spike protein, and the ACE2 receptor, making this interaction a little less efficient. Then Camostat, I mentioned before, the serine protease TMPI-SS2. Camostat has been or has clinical approval in Japan against pancreatitis treatment, and this is targeting the serine protease. And I mentioned to you before, this is similar, a little bit similar to what Tamiflu is doing in influenza infection. So if you give this very early, you could probably reduce or make the entry of the virus less efficient. And as we heard also earlier, the number of viral particles that manage to infect our cells is really critical for the severity of the disease. So reducing the number of particles that make it into the cell might be a good idea. Passive immunization is an old concept that is also used in Ebola outbreaks. Rather difficult to do. It only is protective for a couple of weeks. However, it might be really useful to protect our clinical healthcare personnel for the time being. Epidemics are important on the S protein, soluble ACE2 proteins that really bind to the S protein of the virus and block its interaction with the cell surface receptor, and the IL-6 antibodies, as I mentioned before. And here's just a graphical summary of these treatments. Now, briefly for the vaccines, I see we're running out of time, so I'm trying to speed up a little bit. So there are a number of vaccines currently under evaluation. So here's a figure from a very recent paper in Immunity from Amanat and Kama, this summarizing nicely the different types of vaccines that we have. And the furthest in clinical trials for SARS CoV2 are these RNA vaccines. Now, what are they? Do I have I'm stuck here. Let's see whether I can go forward. No. Okay. I hope you can see the slide okay. I only see half of it. Anyway, so this is the types of vaccines that we have against viruses, and CureVac wait a minute. So the CureVac in Germany, and wait, I see I'm getting a message here. Okay. Okay. I'm not all okay. Good. Fine. So now I don't have the pointer anymore. Anyway, I hope you can see the slide and the pointer. If they give me back the pointer, it would make it easier. Anyway, so there are RNA vaccines that are the closest to completion by a German company and a US company. And the principal idea is that parts of the virus, in generally for CureVac, I know it's the spike protein, mRNA is being generated in large quantities, and then they have a secret recipe to stabilize this RNA. And this RNA is then injected into the patient. The RNA is taken up by the cells, is then translated into spike protein. The spike protein is released, and the antibodies making the patient is making antibodies against this particular protein, and so develops immunity. Now, overall, you can have a look at this table. It's actually quite interesting, summarizing the different concepts. But their final quote of this particular paper is that we're probably too late with vaccine development for this first wave. However, they might be useful if additional waves occur later. And in addition, lessons learned from handling this outbreak will allow us to be better prepared in the future, because the viruses will keep coming, and not only coronaviruses, but others too. So there are lots of lessons to be learned for the healthcare sector and for politicians. Now, just to finish off, some very brief thoughts on global and national impact. We do need to think about co-infections and the role with other viruses. And we need to think about these problems even more so as the virus is now moving into Africa, into South America, where the prevalence of co-infections will be much, much higher. The case fatality rate is, of course, going to be skyrocketing in people who already have tuberculosis or HIV or a number of different infections to go with the virus. So this is something we need to think about. And some of the problems, specific problems that we have in the other continents, where corona is really getting into right now and replicating up to gigantic numbers. So those epidemics will then probably fuel the second wave that we are going to see likely towards end of the year or early next year in Europe as well. However, to put things into perspective and to compare it to other diseases, it's a global tragedy, this virus, and a global challenge for us all. However, we shouldn't forget that other viruses also do a lot of havoc, and other diseases kill millions of people. And on the non-infectious part, we have 10 million people dying of cancer every year. We have 18 million people dying of cardiovascular diseases almost every year. We have 37 million people living with HIV, 50 million died from the Spanish flu, etc. So as much as we need to take care of reducing the spread of coronavirus, we also need to put these things into a larger global perspective, I think. And here are some examples of influenza burden by the CDC. So we have 45 million of cases in the last season, whereas we have 1.6 million of coronavirus cases. Of course, in reality, we have much more cases of coronaviruses that we know of, but still, this is a very large number in comparison. The impact on economy, it's needless to say, we all know that economy is going to be plummeting all over the world. and the dimensions of the crisis are probably not foreseeable for us scientists at this point in time. Unemployment will be rocketing. In the US, I think the US looks really dramatic. The situation in the UK might be equally dramatic once they get their peak. And the UN is calculating that globally close to 24 million jobs could be lost permanently. What the effect on mortality of other diseases, overall violence, social hardship is, we will begin to see after the worst is over. So what will the next 12 or 24 months look like, given the situation that we are all in? As I explained, I think we will have a second wave, and virologists all over the world agree on that. Hopefully a much smaller third wave once a vaccine is there. We will see vaccines becoming available, different vaccines. There will be better treatments for sure. We need to find biomarkers. I'm sure there will be found too, to identify and stratify patients into severe and mild cases in order to also release the healthcare system a little bit. The healthcare systems will have to be adapted to account for such outbreaks. We will hopefully move from social distancing to smart distancing, allowing certain activities in the near future again, at least for us here in Central Europe. The economic impact will be enormous, and this virus is going to have an enormous impact on other diseases, on our social behavior and global interactions. So with that, I'd like to conclude and say that SARS-CoV-2 is actually, from a virological point of view, a classical zoonotic, and highly transmissible RNA virus, which causes a relatively mild disease in the majority of cases, but with a massive and unprecedented impact on us all. And with this, I'd like to thank you for listening. And I have a couple of sources listed here for further information. Yeah, as you know, there's a lot of information out there being updated on a daily basis. All right, I'm happy to take questions if there are any. Okay, there has been some conflicting information on whether ACE inhibitors interfere with the virus adherence to ACE2 protein. Do you know what are the latest findings, if any, exist on this question? So Julien is asking this. And the ACE inhibitors, I think from what I read something yesterday, and indeed, it depends a little bit on the number of viruses and the different inhibitors that are being tested. There are huge differences that they are trying to understand right now. I'm not sure. I think what is for sure that Camostat seems to be reducing the number of uptaken viruses, but this is not preventing viral infections. It's just slowing it down a little bit. And there it depends very much on the timing. And this might be the same for the ACE inhibitors, the timing when this inhibitor is given to a patient and at which stage of the disease the patient is might be critical or account for differences here. Right. Does that answer your question, Julia? What is currently known about immunity? Will a reinfected person be a vector again? Adrian Ries is asking this. Immunity is a very important issue, and I wanted to include it. And I was really looking hard yesterday what is known so far, and there's really not much out there. We know that the virus itself can produce agonists against interferon gamma, and interferon gamma is our key defense of the innate immunity arm. So the virus has some ways to dampen this host interferon gamma response. However, how exactly this works, we don't know. And there's no clear picture on what the specific immune reaction against SARS-CoV-2 looks like. So what I said from this study in Germany that was published yesterday, or the first summary, they said that they have evidence that immunity will last 6 to 18 months. I really don't know how they can conclude this at this point in time, but they did some experiments but didn't disclose it. So we don't know yet. We'll have to wait and see. Hopefully the virus is not changing tremendously, and we will be immune. The ones who have had contact with the virus will be immune during the second wave. If not, we are in trouble again. Ambre Lecomte is asking, for how long is an asymptomatic person considered contagious? Same time as a symptomatic person. Good question. This is a part of the current studies that are being undertaken worldwide. The Solidarity 2 study and also the Luxembourgish Convince study will look at exactly that. We don't know. The asymptomatic individuals probably, in some cases, have a lower viral load, so have been infected with fewer viral particles, and therefore might also not be as infectious for as long as a symptomatic person. But this is really just assumptions, and we'll need to measure this by these randomized prevalence studies that are being undertaken right now. Judith is asking, do you think we're ready to start with an exit strategy? Politicians are thinking of slowly relaxing measurements, but if you look at Wuhan or other Chinese cities, they were in lockdown for much longer, and some cities are still closed. So why would we, after only 4 weeks, be ready to open up shops again? Yes. Well, I think yes and no. We still have to be very careful, and I don't think anything will open up in the after Easter. So we will probably continue our social distancing for a few weeks to come. But if the numbers develop as well as they have been doing so in Luxembourg and Germany, like I said, there is good reason to move into smart distancing. So small shops where people can really stay away from each other, for instance, a little bookshop, and the risk to get infected in such a scenario is very low. And we need to balance our measures against the economic impact. Now, I'm not an economic person, I'm a scientist, but the economic impact here is so tremendous, and that again is having impact on other diseases, on very sick people who don't get the treatment or delayed treatment, and it will have impact on overall poverty, and it will have mortality issues in many areas. So we need to balance this. And in fact, yeah, so I think it should be possible to get into smart distancing and open up some places soon with clear rules, clear distance rules, and then see how it goes. Yeah. Natalie is asking, how high do you think is the herd immunity in Luxembourg, and how will it be possible to increase it to 60% without spiking the curve again? Yes, that is a concern. We probably, I mean, the study in Germany showed that there's a herd immunity of 15%. Now, that is not good enough, seriously not. And this was a place in Germany where they had a serious outbreak. How are we going to get to 60% under the current containment measures? We won't. It's impossible. If we stay at home and nobody gets infected, or very few people get infected, we will not get to 60% herd immunity. So this is exactly the balance that politicians and healthcare officials have to look at the moment, is opening up a little bit, having a controlled, let's say, infection rate, that a number of people, especially the younger, healthy population, does develop immunity and gets in contact with immunity, because that's the best protection the population can have, a higher herd immunity. And then balance this out time-wise until vaccination becomes available. So it's a delicate balance of keeping things closed, keeping us at home, preventing or protecting our healthcare system from complete collapse, but also allowing for some people to actually go out there and probably get infected, but with mild symptoms for the very vast majority of cases. Adrien Ries is asking, what is known about the pathophysiology? Is the problem similar to hepatitis viruses, where the inflammatory reaction does more harm than help? Is there significant epithelial cell destruction? I'm not sure there's a lot of evidence out there. Like I said, over the last 2 days, I really went through the literature, and I couldn't find any detailed information on that. If it comes to the immune system, we're still in the dark. What we do know, what the people in the hospitals can measure, is a cytokine storm. So there's a severe inflammatory reaction, an overreaction in some patients, which is actually doing more harm than it helps. To answer your question, whether there is significant epithelial cell destruction, I can't tell you. Maybe some of the medical doctors or the people treating the patients in the ICUs will know better. But I couldn't find any publications on this in the last few days when I looked. Prashant is asking, what about the mutation of the virus? Is a universal vaccine realistic? Yes, also a good question. It's an RNA virus, like I explained in the beginning. So there is room for mutations. And within the spike protein, I showed you that there are already a couple of mutations that could render a possible vaccine useless. However, with the mRNA technology, they relatively rapidly can develop different vaccines with different sequence background. And I'm pretty sure that this is done right now as we speak. All this information is completely confidential. Even if you write to the companies, they don't tell you anything. But I would assume they do this. They prepare different sequences of spike proteins to be injected maybe together. So similar to what we do with the influenza vaccine, where every year we look at what is circulating, and 3 to 4 different strains with a different antigenic makeup are being combined in one shot. And this is probably what we need to do for SARS corona as well. Alexander Frisch is asking, recently there have been reported several cases of virus transmission from human to animals. Do you think that represents a big risk in the case for our pets too? Do you think that if some of our pets get sick, there would be a chance of transmission from regular pets to humans afterwards, so that it would become somehow a zoonotic loop? Yes, I saw this report on detection in a tiger or something in a zoo. So in a tiger, they detected SARS coronavirus. As far as I know, it has not been found in cats or dogs. So our pets seem to be safe, and the chances are low. But for RNA viruses, it is not impossible. Like I said, species jump can happen. If mutations accumulate, it is possible, but I would say very unlikely. And striking is that a lot of these big epidemics have started in China or in Asia. Why? also the flu epidemics, or the new really dangerous influenza subtypes, because in these markets you have a crowded space where many different animals are in very, very close contact to people. So there the chance of recombination and new subtypes that can make a species jump for this to develop is much higher. So I think living without dogs and cats is not going to be the problem here. Vicky is asking, could the cross-reactivity help with immunity? The cross-reactivity, you mean against the other coronaviruses? It doesn't look like it at the moment. I mean, people have antibodies against these harmless coronaviruses, and they are still not protected from SARS coronavirus 2. However, what we don't know yet is that the people who have antibodies against the other coronaviruses might have, in general, a milder disease. This could theoretically be, and this will also hopefully be an outcome of the global studies on asymptomatic carriers. So we should get an idea about potential cross-reactivity. Jill is asking whether I know anything about the correlation between COVID and new signs of vasculitis. Oh, I haven't seen anything. No, sorry. Not really. Rubens is asking, in Turin, they saw that all the patients with bad prognosis had low levels of vitamin D. Is there any paper regarding a potential role of vitamin D with the disease? Not as I can remember, Rubens. I don't think I saw anything on vitamin D, and I wasn't even aware of this. So maybe you can send me this information later, and I can try and find something. But I didn't see a paper on this. No. All right. No more questions? Or am I stuck here? I can't see any new questions.
I think we are slowly approaching Okay, if there are no more questions, then we will finish this session.
Yes, thank you, everyone.
There's still another question. Wait. Okay.
Nina is asking, how about giving antibodies from cured patients? Yes, I had this on this one slide. This is passive immunity transfer. It's a very old concept. This is possible. It's currently being explored. And as I mentioned, it's a bit difficult because the immunity only lasts for a couple of weeks. And of course, you need to identify enough people who have recovered, who are healthy, who had the disease, and who are willing to give plasma. It is possible, and it's a good idea to protect healthcare personnel in regions that are really critical, where we see in the United States, in England, and many other countries in South America, that the healthcare personnel is not sufficiently protected, and they get sick very easily, and then the system collapses completely. So it would make sense to protect our healthcare professionals with passive immunization. Mm-hmm. Connie Huberti is asking, how are people being chosen for the 1,500 asymptomatic tests? When and where could they be tested? I think this is currently being conducted by some people within this task force. There has been this task force put together in Luxembourg with a number of people from different institutes, and they are currently doing a random selection. They have certain criteria. People have to be above 18 and they have to be apparently healthy, and they will have to be willing to share personal information, etc. And then samples will be taken, I think, in the beginning, and they will look at the viral titer, and then eventually, as soon as serological tests are up and running, they will look at the antibody titers and then follow up the patients over, I think, a full year. And eventually they will also take not only blood samples and swab samples, but stool samples to look at the microbiome and some other samples as well, as far as I know.
All right, that was the last question. So we will close the session now.
Thank you.